My love for developmental biology was triggered 46 years ago in a lecture hall like this one while listening to a lecture by Walter Gehring in Basel. He mentioned that the future germ cells in the Drosophila embryo were specified by cytoplasmic determinants that contained RNA, and that it was not known what these determinants were and how cell fate is specified. There, I found a problem that was approachable and tangible. I wanted to find out what these determinants were. And even if I did not find this out, my love for developmental biology and Drosophila was triggered, and for the next 45 years, I spent much of my life devoted to research on this animal. Drosophila is a powerful model system to understand basic biological mechanisms, such as cell fate specification, because it is easy to keep, reproduces quickly, and we have a vast arsenal of genetic and molecular tools at our disposal. Moreover, these flies look beautiful under the microscope, and they are our distant relatives. The last common ancestor of Drosophila and humans lived some 600 million years ago. This ancestor lived in the sea and looked like a trilobite. That means that already 600 million years ago, evolution with natural selection found solutions to many of the problems that trouble us today. These include how cells divide and grow, and how they make sure that they stop dividing to make our legs and wings the same size, etc. Studies in model organisms like Drosophila from our lab and many others have contributed to the understanding of some of the basic biological mechanisms at the heart of many human diseases. I've been lucky that my interest in how cell fate is specified and how organisms control their size coincided with the productive encounter of genetics, identifying genes by mutations, and molecular biology, cloning genes by characterizing the encoded proteins and their interactions. Using a combination of genetics and molecular biology, we and many other groups were able to identify the genes that control development. For example, during my PhD in Basel, I was able to show how genes that control the number of segments of the body are expressed in stripes of cells in the early embryo. During my postdoc in Berkeley, I turned to the beautiful compound eye of Drosophila and could characterize the gene that controls the fate of the R7 photoreceptor cell. Cloning and characterizing this gene was sufficient to get me a position as an assistant professor at the University of Zurich, where we continued our work, now with a team of talented graduate students. Above all, Konrad Basler, Damian Brunner and Barry Dixon, to decipher the signaling pathway that determines the fate of photoreceptors in the Drosophila eye. This evolutionary conserved Ras-MAP kinase signaling pathway is involved in cancer in humans, and by using genetic tools, we were able to identify the functionally important components of this pathway, many of which are now used as drug targets against cancer. After about 10 years, we turned our attention to the question of how genes specify the size of cells and body parts. Again, this change was triggered by a chance discovery by Juan Riesco, a postdoc in the lab. He discovered a small fly mutant he called Chico. Even though I dismissed it at first as just simply a starved fly, he followed up the characterization and convinced me of this new research direction, which actually continues to fascinate me and my group to this day. it turned out that we discovered another conserved signaling pathway, the insulin signaling pathway, involved in growth control. So far, we always looked at the role of single genes on developmental decisions. When I joined the Institute of Molecular Systems Biology at ETH and interacted more with my friend and colleague Rudi Abersold, we realized that natural selection does not act on single genes, but on phenotypes, and these are produced by the interaction of the genome with the environment. Now we had access to the tools to address this broader question. Sibyl Fonash, a very talented graduate student, and 2 postdocs, Hiro Okada and Ryohei Yagi, took on this task. We were able to begin to analyze how cell size is controlled at the level of the genome, the transcriptome, and the proteome. We even built a fly catwalk to automatically sort hundreds of flies based on their wing size. Even though all these research activities come to a close now, I'm very happy that we can use our knowledge on Drosophila in a project for the health of honeybees in collaboration with Agroscope in Bern and the University of Lausanne. With the advent of genome sequencing getting less expensive and private companies like 23andMe moving into the space of direct-to-consumer genetic testing, I realized that we humans are better model organisms to study health and disease than flies. Moreover, we can talk and go to the doctor and describe our phenotypes. Moreover, with our smartphones, we can record data continuously related to our health and actively contribute it to research. This is why I offered my family their personal genome analysis as a Christmas present 12 years ago, and together with the bioethicist Effie Vallena, we conducted research on how scientists and students received their personal genome information. The deepening of the understanding of personalized medicine and health requires the combination of different categories of health data of millions of people. For this, people have to seize control over the use of their data, rather giving their data to internet companies for free data services via apps. For the past 10 years, I have been advocating that data banks, organized as citizen-controlled cooperatives, in which we control access to copies of our personal data, in the same way we manage our money, would be a way towards a fair and democratized data economy as an alternative to the surveillance capitalism model of the US or the Chinese state surveillance model. Helping projects towards this goal will certainly continue to occupy me in the future. In 2004, we organized the exhibition Der gespiegelte Mensch in den Genen lesen in the Landesmuseum. For me, the translation and communication of research has always been an important part. Therefore, I helped to found Life Science Zurich, a joint initiative of the University of Zurich and ETH. In 1999, I founded, together with Konrad Basler, Michel Aguet and Mario Jenny, the genetics company, with the aim to discover new drug targets with the help of Drosophila genetics. At that time, simply the fact that 3 professors wanted to start a biotech company was sufficient to obtain venture capital. 35 million francs and several strategy changes later, we and our investors had to realize that a company that focuses at the very beginning of the 15-year drug development cycle is not a viable business model. While our company was not successful, the Biotechnopark in Schlieren, where our company was located, is a super success story. 55 companies, 1,100 jobs, 55,000 square meters, and over 3 billion Swiss francs of investments. Even though I'm the president of the Biotechnopark, its success has to do with a combination of a visionary real estate entrepreneur, the late Leo Krummenacher, the leadership of its CEO, Mario Jenny, and the numerous startups from ETH and university. Obviously, less successful was my engagement in university management. I acted as president of ETH in 2005 for just over a year. Lack of experience, impatience, and also lack of support from above led to a slowing down of the innovation process of research and teaching. I'm sorry that I carry some of the burden of this setback. On the other hand, through the presidency, I obtained a professorship at ETH and had a chance to experience ETH from the side of a professor. The change to ETH also triggered my interest in the education of students and teachers. I finally realized that in the design of lectures, I oriented myself on the role models during my own education. While we are constantly adapting our research to the latest developments, in teaching we orient ourselves to the distant past. Moreover, while we do research in teams, we design lectures mostly alone. Eliciting the help of my postdoc Katja Köhler in designing lectures and curricula suddenly made teaching interactive and fun. We were able to start the Center for Active Learning at the Department of Biology. CAL is a unique entity in which senior biologists assist professors in making lectures more interactive. The results show that students learn more and they appreciate this more active learning process. Education and teaching can be fun. We were able to help students to think beyond the passing of exams and to see their full potential as active agents in society and research. With the event Your Future in Biology, we were able to show biology students the different career trajectories people took after their studies. With the block courses on bioentrepreneurship and biological approaches to sustainable development, we could tell students to see themselves not merely as students, but as change makers. And finally, in facultative book clubs for first semester students, we were able to engage willing students in discussions on the historical and societal implications of biological research on the basis of popular science books. It has been a lot of fun. Looking back over my time as professor at ETH, I appreciate the privileged position that we are in and the potential of basic research and its application to real-world problems, including climate change, health, and disease. I wish that the barriers for collaboration between departments and institutions could be lowered further, and that the movement toward open science and free access to data could be increased. In education, I wished that creative thinking, teamwork, and working on real-world problems could find a larger space already during early semesters at ETH. Finally, my work over the past 10 years has shown the largely untapped potential of personal data being controlled by the individuals. This does not only relate to health data, but also to personal data related to education. ETH could take a lead in the movement towards personalized education before this space is taken over by American companies. In summary, it has been an exciting journey in the land of academia. I realize how privileged I have been to choose my own research direction and to follow it with an ever-changing team of motivated and talented students and postdocs. I would like to express my gratitude to all the institutions that supported me over the years, starting with the University of Basel, UC Berkeley, the University of Zurich, ETH, and the Swiss National Science Foundation that supported much of my research. I will not mention any names anymore now, but will do this in written form at the end. And finally, I'd really like to thank all my colleagues at DBio and IMSB and all the administrative staff. It has been really exciting working with you. At the end, I would like to thank my wife, Eva, and our sons, Raphael, Timothy, and Robin, for being tolerant and patient for my frequent absences as a husband and as a father. And now, the great divide is calling.